Yes, so I hope you guys have seen the videos of week 5. So, as our session goes always, we'll first have a recap of what you guys must have studied in week 5, and then we'll take the doubts. Okay, so let's start. In this week, what we actually studied is yes, in this week we studied about the replication part. What is replication? If you guys remember, in week 4 we studied about this central dogma of biology. So in central dogma of biology, I told you about the processes that happens in the central dogma. So the processes that happens there are replication, transcription, and translation. So, replication is the process in which a DNA is forming a copy of itself. The process of formation of a copy of DNA is called as the replication. Okay? So, if the life must proceed in any sense, then it should have a copy of the genetic material. Otherwise, how will it proceed? Where from the information come that what protein should we make, what should be the process for different kind of chemical reactions going on. So yes, it should have a copy of the genetic material inside the daughter cell also, so that it can provide the information required for daughter cell to proceed the life. Okay, so this is what we studied. It is the replication. So DNA replication is a biological process that helps to transmit DNA, that is genetic information, from generation to generation by producing two identical DNA strands of daughter cells from the double stranded parental DNA. ओके, सो डबल स्ट्रैंडेड पेरेंटल डीएनए जो होता है, उससे डॉटर सेल्स के लिए इट मेक्स द टू आइडेंटिकल डीएनए स्ट्रैंड्स. ओके, now the importance of replication is a way of duplicating all the genetic information from all living parent organisms to daughter organisms, thereby helping maintain the genetic materials intactness and the organism's survival, basically to survive, because it has all the information a cell requires to proceed in the life. Okay. So we'll proceed towards now in we as we discussed in the initial lecture only, that we have two types of cell, the prokaryotic cells and the eukaryotic cells. So obviously replication, because both the cells have much more differences. Prokaryotes do not have a well defined nucleus in which the genetic material stays. They have a single chromosome, while the eukaryotes have a defined nucleus. They have many chromosomes, not a single chromosome, depending upon different species, the complexities of a species. So obviously, as the complexity is different, as the cells are different, so obviously there will be a difference in the process of replication also. The process of replication will be different, somewhat different. The overall process has both the cell types have overall similar stages, but the enzymes involved is different, and somewhat a minor difference in the process also. Okay, so first of all, we'll proceed with the prokaryotic genetic material. So prokaryotes, as I have already told you, they have a single chromosome. A prokaryotic genome can mostly be found in the cytoplasm because they do not have a well defined nucleus. They are negatively supercoiled and circular. Generally, they can be found in singular number. However, there are some exceptions where the number of the genome is higher. That is, in Vibrio cholerae, it is 2. In prokaryotes, for an example, E. coli, they contain a self replicating extracellular genetic material. This is an extra genetic material, we can say, they have, that provides some additional benefits to these prokaryotic cells, which we call it as plasmids. Okay, so what are plasmids? Plasmids are only up to 1500 to 2000 kb, not much larger, therefore very small compared to the actual prokaryotic DNA, which is in MBs. Okay, they contain origin of replication, hence it replicates independently. They do not require the machinery of the cell to replicate. They have their own origin of replication, So they can divide on their own, they do not require machinery from the cell itself. Okay. So features of prokaryotic replication. Uh, uh, Before going into the details of the features, you guys must understand this thing, that mode of replication. What is the mode of replication for the genetic material? So initially it was proposed three modes of replication. Three ways it was thought to be that the DNA is replicating. One was conservative, second was semi-conservative, and third was dispersive. So what happened in conservative? It says that the two DNA helices are produced from one original DNA helix. One helix contains downright parental DNA. That means no new DNA additions is there. The original parental DNA is as such. ठीक है? और दूसरा is semi conservative mode. In what happens in semi conservative mode? It shows that the daughter DNA is being formed from the parental DNA itself, but it is not like DNA of parents is getting separated wholly and the daughter DNA is altogether the new DNA. No, it is not like that. It will be semi conserved. You'll see it, you'll get it clarified from the diagram I'll show you guys. Next is the dispersive mode, which shows too much mixing. That means some pieces are of parental DNA, some pieces are of the daughter DNA. So that is also not possible. Significant that can cause a lot of mutations and problems. So that is also something which is not possible. So as the diagram says, in conservative, the parental DNA is remained as such, and the replicated DNA is making a whole new DNA for the daughter cells. It is not like mixing is happening, which happens in semi conservative mode, in which one strand is from parental DNA and one strand is from daughter DNA. So this way information is also conserved and new information is also getting transferred to the daughter cells. In the dispersive mode, what happens is, in the dispersive mode, it shows half part, some parts are of parental DNA, some parts are from the daughter DNA. It is not something ki strand wise, it is pieces wise distribution, so that is also not possible. Okay, and this was proved by what? It was proved by Michelson and Stahl experiment. What they did is, they took two medias to grow the E. coli cells, one having the nitrogen isotope N14 and another having the nitrogen isotope N15. So, as we all know, nitrogen is a very important compound for the nucleic acid because it forms the nitrogenous bases, which are actually the one which are carrying the information. So they took nitrogen isotopes because that will be a short experiment. So they grew the E. coli cells in both of these medias, and they found out that the cells is actually dividing by semi-conservative mode of replication. Okay, so this was what they got. Next is, now we have got an idea of what is the mode of replication for the genetic material. Now we'll further proceed towards the process of replication. So before going into the detailed process of replication, there are some terms you must be familiar with. One of them is replicon. What is a replicon? So replicon is actually a DNA fragment of prokaryotes that undergoes replication. Okay, it has origin of replication at terminus region also, because it should be told that where to start and where to end. If you don't tell any genetic material this information, then it will be a enormous process. It will keep on going and going and going. So that is why it should have an origin of replication and also an terminus region. For example, E. coli has one replicon on its genome. That means it has only single origin of replication for whole of its genome and a single terminus region for whole of its genome. Okay. Eukaryotes, on contrary, has multiple origin of replications, and therefore they are called as multi-repliconic. Okay, so monorepliconic and multi-repliconic, two terms are there that you guys must be familiar with. Next, what is origin of replication? So at the names, as the name suggests, the origin of replication represents the starting point of replication in prokaryotes, where from it will this is that point where it gets the signal that yes, you can start your replication from here only. It gets its initial sorry, initial signal on origin of replication. It gets all of the components required to start the replication there and then only on origin of replication. So that is why origin of replication represents the starting point of replication in prokaryotes. It is approximately 245 base pairs long and it is 80 rich. Okay, so these are two peculiar feature of origin of replication for prokaryotic cells I'm speaking. So it is 245 base pairs long and it is 80 rich. It is a cis acting sequence. That means they can affect only on the molecule of DNA on which they reside. It will not affect the molecule of DNA which the other DNA molecule. Basically, it will only affect the DNA molecule in which they are present. Obviously, the sequence is present in a particular genome, so why it will affect the genome of the other genome part where it is not present. Okay, so why 80th sequence is preferable for melting unwinding of duplex DNA? Because as we all know, 80 complementation has two hydrogen bonds while the GC complementation has triple hydrogen bonds. So it is easier to melt two hydrogen bonds than three hydrogen bonds. So that's why it is 80 rich. Less energy is required to break down the hydrogen bonding than GC rich sequencing. Okay, so this is what was the origin of replication. And the melting of duplex DNA is ATP hydrolysis dependent, as the energy released helps break the hydrogen bonds between A and T. So it requires energy to start the DNA, the replication, and obviously the melting of the duplex DNA. Okay, above mentioned 80 rich sequences are recognized by the enzyme DNA helicase to initiate the unwinding process. So helicase means something which is breaking the helix arrangement. So that is why the name DNA helicase. Okay. So this is just to show you what is origin of replication, what is there in the origin of replication. So it has two regions on that: duplex unwinding element and DNA oligomerization region. If you can see DOR and DUE. So DUE contains 13 mer repeats or sequences. Three sequences are there, while the DOR contains 9 mer repeats and 5 such kind of repeats, 5 copies of such kind of a repeats is there in the DOR. And the whole of this along with some other sequences side by side makes the origin of replication. Okay, so origin of replication of E. coli is known as Orice. It contains two short repeat motifs, which has 5 copies of 9 mer sequences, 9 base pair sequence you can say, and 3 copies of 13 base pair sequences, which are again 80 rich repeats, where DNA starts unwinding and other the DNA polymerase and other enzymes can come and bind so that replication can be started and proceeded. Upon binding of DNA at the 9 mer range, the 13 mer range starts melting. So this is where the process of starting of DNA replication is coming into the picture. So whenever an enzyme named DNA binds to the 9 mer range, the 13 mer range starts melting. Okay, and the Orice contains 11 copies of 5 prime to 3 prime GATC repeats, methylated on adenine on both the strands. ठीक है, तो ये वाले repeats भी present होते हैं इसी में origin of replication में. ठीक है, only complete methylation can lead to the initiation of replication. If it is semi methylated, then it won't lead to the origin of replication. Only the complete methylation of these sequences of the adenine in these sequences will lead to the origin of replication. ठीक है। उसके बाद replication fork. Now what is replication fork? So this is the y shaped structure. If you can see it in the picture, the as the replication starts, it proceeds in both the directions. So this is a bidirectional replication process, and when it is proceeding in both the direction, it is forming somewhat y like structure on both the ends wherever it is proceeding. If you can see it on my cursor, so wherever it is proceeding, it is showing Y type of structures. So this is called as the replication fork. The Y-shaped structure is generally found when the DNA starts melting and opening up. As the DNA opens up bidirectionally at the origin of replication region, two replication forks are generated because it has started in both the directions. Okay, an extension of two oppositely directed replication forks lead to a replication bubble. This whole bubble like structure is called as replication bubble. Okay, and to maintain the single strand situation. Now single strand will be unstable obviously, because it will try to hydrogen bond with the complementary nitrogenous base on the both the strands. So to stabilize that, single strand binding proteins, which are also called as SSBs, they coat the single strands of DNA to stabilize it and to prevent it from rewinding. Okay. So here are some of the important enzymes which are important for prokaryotic replication. Helicase, as you guys know, to open the helix. SSB is just to stabilize the single strands of DNA. Topoisomerase, they work at the region ahead of the replication, basically to release the tension. If you take a rope which is made up of like some mini ropes, and they are wound around each other, so if you will try to open that winding, obviously it will develop some tension in the extended region of itself, right? So this is how the topoisomerase is helping here, by releasing that tension so that it won't break the DNA and it won't affect the structure of the DNA. Okay, so it prevents the supercoiling and it helps in unwinding of the region ahead of the replication fork. Next is, sorry, DNA polymerase 1. This is the first DNA polymerase to be discovered in E. coli by the Nobel Prize winner Arthur Kornberg. ठीक है. Structural gene name is Pol A. This is a monomeric protein and the activity is 5 prime to 3 prime polymerase activity. This is its speciality. Okay. So this will be the main enzyme which will be helping in the replication. Sorry. Screen is shared, right? Just a moment, guys. Yes, it is shared. Hmm. Next is this is still the continuation of DNA polymerase 1. So 5 prime to 3 prime exonuclease activity is independent of other activities. Now, the protease can cleave this polymerase 1 into two fragments, like a larger C terminal clinov fragment and the smaller fragment for proofreading activity. Okay. One of the very important information about DNA polymerase 1. Now DNA polymerase 2, it is the monomeric protein with Pol B as the structural gene. Structural gene name is Pol B. Okay. So activity इसकी भी है 5 prime to 3 prime polymerase activity. Exonuclease activity is 3 prime to 5 prime. Low polymerization rate, low processivity rate. That is why it is generally used in the repair mechanism but not as the main DNA polymerase to replicate the DNA. Now DNA polymerase 3, it is the primary DNA replicase. So yes, it is the primary DNA replicase with the structural gene name POLC, multi-protein complex of 10 different polypeptide subunits, high polymerization rate, high processivity rate. DNA polymerase 3 stays as an whole enzyme. during replication. So DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. ठीक है. So DNA polymerase was the first enzyme DNA polymerase discovered in E. coli. 5 prime to 3 prime polymerase activity होती है इसकी. ठीक है. एक्सोन्यूक्लियस एक्टिविटी भी होती है 5' to 3'. प्रोसेसिविटी लो होती है, लो पॉलीमराइजेशन रेट होता है, तो इट इज नॉट द मोस्ट प्रेफर्ड डीएनए पॉलीमरेज, ऑल्दो इट कैन ऑल्सो हेल्प बट इट इज नॉट द मोस्ट प्रेफर्ड. डीएनए पॉलीमरेज 2 सेम प्रॉब्लम, लो प्रोसेसिविटी रेट, लो पॉलीमराइजेशन. So it is also it is actually involved in DNA repair. DNA polymerase 3 has the highest processivity and high polymerization rate. So that why it is preferred for DNA replication. Name of the enzymes still continues. So it is just showing you the subunits of DNA polymerase 3. We're not going into the details. You must have read it from the lecture. Okay. Next is DNA polymerase 4 and DNA polymerase 5. Both are Y family polymerases that don't have 3' to 5' exonuclease activity, low catalytic efficiency, low processivity and fidelity. Okay. उसके बाद डीएनए प्राइमेज इज रिक्वायर्ड टू सिंथेसाइज द प्राइमर्स that will be helping in legging strand formation. And DNA ligase, they'll actually seal all the nicks or gaps in the DNA. So DNA ligases are to reseal the DNA wherever there are some nicks or some missing base pair or something. After repair, it will reseal the DNA. Okay, so the stages of the prokaryotic replication, as I've told you: initiation, elongation, termination. Initiation is the recognition and starting of replication. Elongation is the replication fork that leads the DNTP synthesis and proofreading. Basically, the elongation means the complete replication, the forward moving of the replication process is elongation. Termination means the end of the replication, it is the entirely stopping of the replication. So E. coli chromosomal DNA is circular with no free ends and is replicated bidirectionally, therefore it resembles the Greek letter theta. Hence, this replication mode is known as theta replication, which is another name for prokaryotic replication. I'm telling you again. It can be seen in the gram-negative bacteria. Some commonly used plasmids like call even RK2F and P1 bacteriophages as well. So it is seen mostly in the gram-negative bacteria. Steps of initiation, I have told you. DNA A protein will bind to 9-mer, which will lead to the melting of the 13-mer AT-rich region. And then further melting is carried out by recruiting hexamer protein helicase DNA. When DNAs will when the DNA helicase will open up the helix of the DNA, then SSBs will come into action. They will help in stabilizing the single strand of the DNA. Okay, so this is whole of the process of the initiation. We are again not going into the details. Then the stage of elongation comes. What happens here is DNA B helicase separates two double helix DNA strands binding to the lagging strand template at the replication fork and moving along according to the polarity, that is 5' to 3'. Okay, DNA G primase का क्या काम होगा यहाँ पे? Sorry. Yes, DNA G primase का क्या काम होगा? DNA G primase associates with DNA B and synthesizes RNA primers. RNA primers are required to make the Okazaki fragments or lagging strand. Okay, lagging strand की formation के लिए हमें Okazaki fragments की जरूरत पड़ेगी, because the 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 other band that is having the leading strand in which the replication is proceeding from 5' to 3', it can easily move in the direction, it can start and it can move from the single primer binding. But for the lagging strand we have to again and again put some primers so that an enzyme can get a place to bind and place where the replication can start. So it is generally for the lagging strand we need this primers of to make these Okazaki fragments, and later on these Okazaki fragments are joined by the ligase enzyme. Okay. So DNA polymerase 1 removes the RNA primer of both the strands by 5' to 3' exonuclease activity, which generates a gap after primer removal in both the strands. Okay, primer remove karenge because these are RNA primers, not DNA primers. So they must be removed. So once they are removed, then the gap must be filled. So DNA polymerase helps in filling of these gaps, and later on DNA ligase will come to join the two fragments. Okay. Next is the role of gyrase. Gyrase का क्या role है? Due to unwinding with the help of DNA helicase, the ds-DNA in front of fork become positively supercoiled. If the supercoil increases, the fork will halt, and therefore to overcome the halting from supercoiling, ये लोग क्या करते हैं बीच में, they make some nicks so that the tension and the supercoiling can be released, and then the replication of DNA can proceed smoothly. ओके, प्रूफ रीडिंग एक्टिविटी का क्या काम होता है? सो डीएनए रेप्लिकेशन इज अमेजिंगली एक्यूरेट with only one error in 1 billion nucleotides incorporated. So it is very less, but yes, sometimes the mistakes do happen, sometimes due to environmental factors and many factors. What happens is either cell skips a nucleotide, or sometimes it can put a wrong nucleotide in that place. So in that case, proofreading activity helps. After the initiation and elongation, termination must happen. So as there is a bidirectional replication, the forks will meet at a position dramatically opposite to the origin of replication on the genome, and the termination region contains multiple copies of about 23 base pair long sequences. ठीक है? And every terminus or ter sequence acts as the recognition site of terminus utilization substrate. So this allows one replication fork to pass if its movement is in the one direction, but blocks the progress if it is from the opposite direction, and the directionality depends on the TUS protein localization in DNA helix. Okay, so TUS has a very important role. After the replication process, two new circles are physically interlocked or catenated. This decantation is carried out by the topoisomerase 4 to generate separated two daughter double-stranded DNA, because we all know that topoisomerase 4 was working where? टेबल में देखा था हम लोगों ने. Yes, it helps the DNA to, it helps actually, it is not like ki it is mainly the enzymes for DNA replication, but it is involved in the translation, synthesis and replicating damaged DNA by bypassing nucleotides that can block the progression of replication fork. Basically it checks all the DNA that whether it is fit to proceed further, or is there some kind of a problem in the DNA. Okay, so this was the prokaryotic replication. There are other types also involved for prokaryotic DNA replication, that is the rolling circle replication and the D loop replication. Just give me. So the rolling circle replication, it is found in archaebacteria, bacteriophage, plasmids, viral DNA, and viral RNAs. So it, what happens is the DNA polymerase keeps on revolving around one strand of this around both the strands of this genetic material, making it a rolling circle type of replication. ठीक है. Next, as I told you, it is a D loop type of replication in which one part of this plasmid which needs to be replicated is protruded out, and the replication happens there. Okay, so the application is found, it happens in a single direction. It is found in small circular and organellar DNA. which where one triple stranded structure called as the displacement loop is formed. Okay, तो माइटोकॉन्ड्रिया में मिलती है और क्लोरोप्लास्ट में भी मिलती है. It is found in the organelles. Next is the eukaryotic replication of DNA. How the DNA is replicated inside the eukaryotic cell. So we all know what is replication. We have just discussed it like 10 minutes back. Important difference between prokaryotic and eukaryotic replications are. What are the differences? Eukaryotic DNA is larger than prokaryotic DNA. One thing, as I've already told you, it is in MBs. Okay, the eukaryotic DNA movement of fork is slower in eukaryotes than prokaryotes. So basically, the Y fork, as I've told you, is slower in case of eukaryotes, but it is higher in case of prokaryotes because because nucleotide have to disassemble so that DNA become available for DNA polymerase. Sorry. Distinct package of eukaryotic DNA in terms of chromatin. Okay, so distinct package of eukaryotic DNA in terms of chromatin, because we all know they have a well defined nucleus while the prokaryotes do not have such nucleus. Okay, what is the replication rate? So replication rate in case of prokaryotes is an E. coli replication for progresses at approximately 10,000 base pairs per second, while in case of replication rate is 10 times slower than prokaryotes, that is 50 nucleotides per second. So this much difference of replication happens in prokaryotes and eukaryotes. These are different types of the DNA polymerase present in eukaryotes, that is alpha, beta, gamma, delta, epsilon. Five types of DNA polymerases are present in case of humans, although in prokaryotes also it was five types of DNA replicases, DNA polymerases, sorry. So the properties are intracellular compartment, which is nucleus, which is, sorry, present for alpha, beta, uh, which is uh, which is uh, which is important for the replication in case of eukaryotes, and for alpha beta it is nucleus where the replication will happen. In case of gamma it is mitochondria because it is an organellar DNA polymerase. Gamma and delta, delta and epsilon again nucleus. Same goes with their biological function and all stuff. We are not again going into the details, you can read it from your slides. Now, the DNA polymerase delta is what we are talking about. It is located in the nucleus, catalyzes the synthesis of, lagging strand. The high processivity when interacting with the proliferating cell nuclear antigen. It is also associated with the helicase activity. Okay, so it is generally involved in the synthesis of the lagging strand, not the leading strand. DNA polymerase epsilon, this is located in the nucleus, and again they catalyzes the repair mechanism. It also catalyzes the removal of primer and filling the primer gap in Okazaki fragment. So it has two functions. It catalyzes the repair mechanism and it helps in the formation of the Okazaki fragments. Next is the replication factor C, also known as clamp loader or matchmaker. So, assist in DNA polymerase delta to form clamp between DNA and pcDNA, pCNA, and this plays an important role in selling up a link between DNA polymerase and DNA polymerase alpha, so that loading and lagging strand synthesis take place simultaneously. Okay, so it is the clamp loader, or we can say is the part where the recognition will happen. The clamp will be loaded. After the loading of the clamp, the another part of recognition will happen, and then after the DNA polymerase will come and start the replication. Okay. Now the histone dissociation and association. If you guys remember last week's lecture, so you guys must be knowing that uh, in the last lecture we discussed about the packaging of the chromatin. Okay, and for that packaging we required an octamer, and that octamer was histidine. Sorry, histones. Sorry. It is histones. So histones are actually the basic or are the basic proteins that helps in winding of the DNA around itself, and then after winding it will make the whole arrangement, the arrangement of this DNA in the form of the chromosomes. So the whole process through which the DNA will go through and ultimately will be stable as chromosomes inside the nucleus will be because of the presence of histones around which the DNA is actually stays as in the coiled form. Okay, so obviously this histone protein must be removed or kept at side for some time when the replication process is going on. So this is what happens in case of the dissociation of histones and synthesis of histones. His synthesis occurs simultaneously with DNA replication, while the dissociation, what happens is methylation at the fifth position of cysteine residues by a DNA methylase transferase appears to functioning by losing up the chromatin structure, because stones must be given some signals that yes, it is the time, you please release the DNA part so that it can get replicated. Okay, so before initiation, there happens is the pre-initiation phase. What happens is in the pre-initiation step is the crucial step that prepares like DNA for the actual replication process. This steps primarily occurs at origin of replication, which are specific DNA sequences. The process of identifying these sequences is known as the replicator selection. Okay, and this process leads to assembly of multi-protein complex at each replicator in the genome. So it is a very important process. You can't enter the initiation process directly in case of eukaryotes. You must clear the pre-replication phase. Okay, so this is what all has been written here. You guys can read it, and still you guys feel any confusion, you can ask me. After all the enzymes got loaded on the DNA and all the enzymes have taken the position, has provided the signal key yes, now the DNA is safe for replication, and obviously we have all the nutrients and other stuff. So that then the initiation process starts. It involves the coordinated action of various protein complexes and enzyme to ensure accurate and faithful duplication of genetic material. This process ensures that each daughter cell receives a complete copy of genome during cell division. अगर किसी भी daughter cell को if they get not the full set or some defected set, then it can lead to the death of the organisms also. Okay, sorry. So ARS is the autonomously replicating sequences or replicators. They are present in yeast. Specific site for initiation, or they are present in most of the cells. They are the specific site for initiation of the DNA replication. ठीक है. So this is all about ARS. You guys can read it. Replication fork में you guys know कि क्या होता है. Four components are there: DNA helicase, DNA polymerase alpha, DNA और उसके बाद single strand binding proteins and RFA. Okay. So you guys know the function of all of these enzymes. Next is the two additional factors which play important role in the application of eukaryotes: pCNA and RFC. RFC is the clamp loader and pCNA is the proliferating cell nuclear antigen. It is the one which actually acts as a clamp to keep DNA polymerase delta to keep dissociating of the leading strand and thus increasing the processing of DNA polymerase epsilon. ठीक है, तो this is one of the important components. Then after comes the process of elongation. Now initiation has happened. The next step should be elongation only, where the actual replication will happen, the replication process will happen. So this is that phase where the complementary nucleotide bases will be added on and the whole sequence will be replicated. Okay, so this was about the replication only. uh, Not particularly replication, there are many things we are going to discuss further. Now there comes an enzyme primase. What is the name suggest? Primase means that is something which can digest the RNA primers that we need for the lagging strand formation. So all newly synthesized polynucleotide strands must be initiated by specialized RNA polymerase called as the primase. ठीक है, so जितने भी RNA polymerases हैं, they must be initiated by primase. RNA polymerases will help in the formation of the primers. ठीक है. Yes. It initiates polynucleotide synthesis by creating a short RNA polynucleotide strand complementary to the template DNA strand. Okay. So it is something which is DNA dependent RNA polymerase. You guys must remember this term. These are DNA dependent RNA polymerase which are actually helping in formation of the short segments of DNA, so that the replication process can start at the lagging strand. Okay. The leading and the lagging strand, as I've been using this term quite some time. So what is leading strand and what is lagging strand? Leading strand is the one, it is synthesized continuously towards the replication fork as the helicase unwinds the template double stranded DNA. While in case of lagging strand, as I've told you, the small fragments that it forms are called as the Okazaki fragments. It is synthesized in the direction away from replication fork and away from DNA helicase unwinds. This strand is synthesized in pieces known as the Okazaki fragments, and other fragments begin with its own RNA primer. It needs something, the lagging strand, to start the replication. So, what does DNA polymerase delta and DNA polymerase epsilon do together? So, looping of the lagging strand allows a combined polymerase delta and polymerase epsilon asymmetric dimer to assemble and elongate both the leading and lagging strands in the same overall direction of the fork movement. So, actually, the formation of after the formation of the Okazaki fragments, the stabilization and assemblance of all of these fragments to form a single strand and other processes, all of these processes are handled by DNA polymerase delta and DNA polymerase epsilon. Okay, next for last step is the termination. When the replication forks meets each other, then the termination occurs. It will result in the formation of two duplex DNA. Even though the replication is terminated, 5' end of the telomeric part of newly synthesized DNA found to have shorter DNA strands than the template parent strand. Okay, so here comes a new enzyme, that is telomerase. So, first of all, what are telomers? Telomers are actually the ends of the chromosome. Okay, which keeps on getting shortened upon division, because these are the actually repeats region, and these regions are not fully replicated when we are replicating the DNA or when the chromosome is being replicated. Okay, so during eukaryotic DNA replication, telomeres play a crucial role in ensuring the accurate replication of the linear chromosome. Eukaryotic cells use a semi conservative mechanism to duplicate their DNA, and this process poses a challenge at the end of the linear chromosomes. Telomeres serves to overcome this challenge and maintain the integrity of the genetic material by presenting at the ends of the chromosome, where they are actually GC rich sequences. They are tandem repeats, and they are generally 5 to 8 base pair repeats. Okay, so linear genome, including those of several viruses as well as chromosomes of eukaryotic cells, poses a special problem completion of replication of the lagging strand uh, uh, because of the presence of the primers and other enzymes that are present. If the DNA could not be replicated, the chromosome would shorten a bit with each. Uh, so what happens is, as I've told you, ki all the telomeres that are present at the ends of the chromosome are not replicated fully. Whenever a chromosome or DNA is replicated, so for that telomerase plays the important role. It helps in the replication of these telomeres so that the chromosome won't be damaged, because these telomeres actually provide a protection kind of thing to the edges of the chromosomes. Okay, so the regeneration of telomeres, what kind of a repeats are present in which organism is what has been shown here. Function of telomerases: telomerase uses its own RNA component which is present in the telomerase itself as template and parental DNA as a primer, and then it will form a strand complementary to the RNA component that is present in itself to elongate the telomere so that it doesn't shorten with each replication. Okay, next is the thing is the DNA damage. So we have completed, we have learned the replication part, we have studied that part. Now the next thing that comes in the discussion is the DNA damage, which is a very I would say an important phenomena when you are replicating the DNA, when you are studying about replication, when you are studying about cell cycle, when you are studying about the expression system, anything. So you guys must know this thing, that the DNA damage is a very important aspect of that, because it happens. And when the DNA damage is happening, then it can lead to very serious issues. Sometimes, some kind of a deletion is happening, then it can lead to the uh, gene disruption, because you won't be having the full gene and the full expression of the protein, and obviously the function of that protein will be hampered, and so that the function that protein performs in the cell. Same goes if there is some kind of point mutation, a base pair is changing, then also it can lead to some kind of serious issues sometimes. And if some additional sequences getting added in somewhere inside the genome, so there are many kind of damages that can be seen in case of DNA while getting replicated and all. So you must know what kind of a damages does DNA faces. Okay, so it is known that genome is not a static entity. Hence, it is highly subjected to variety of heritability changes. A sudden change in the sequence of an organism's genome that give rise to alternate form of any gene is called as what? Mutation. The right term for that is mutation. As a result, DNA gets damaged. And if the DNA is damaged, then it will make faulty proteins, and these faulty proteins will lead to faulty expressions. Okay, these mutations are mostly recessive and lethal. As I already told you, they are recessive and they are lethal. Mutations are random and can occur anytime in any of the cell of an organism. It is not like that if a mutation occurs once, the same mutation cannot occur again. Mutation is recurrent, that means they can come again and again. How does DNA gets damaged? So on the molecular basis, DNA can be damaged in two ways, either spontaneous or the other ways through mutagens. Some induction is there, some agent is there due to which they can get mutated. So the spontaneous damage occurs without treatment of organism with any exogenous mutagen. No external mutagen is present. It is from within itself, like some base pair has changed, some faulty base pair has been put on the place of the right, like if the base pairing is AT, and while getting replicated, instead of T, the system puts C there, then it can lead to a point mutation, a change in the DNA. Induced mutation arises when mutagens react with the parent DNA, which causes structural alterations in base pairing. Okay, so agents जो mutagens के हो सकते हैं which increases the frequency of mutation. Those are called as the mutagens in case of the induced mutation, obviously. So that agents that can cause DNA damage. In case of spontaneous mutation, what can they be? Error in DNA replication, spontaneous lesions, transpositions like tautomeric shift, depurination, and depyrimidation. There are many kind of modified sequence, modified nucleotide base nucleotides are also present, which can also lead to some kind of a point mutation. Now, the induced mutation, what happens there is the chemical mutagen or the physical mutagen. Both are involved in induced mutation. For example, the chemical mutagen are alkylating agent, deaminating agent, base analogs, intercalating agents, while the physical ones are UV radiations, ionizing radiations, heat. All of these kind of things can lead to some kind of a mutation in the DNA. Spontaneous mutation, that means the tautomeric shift. The stable keto forms of thymine and adenine and amino forms of adenine and cytosine undergoes tautomeric shift to form the unstable enol and imino forms respectively, which can lead to the change in the base pairing and the point mutation. Okay, next is the substitution mutation. That means substitution of one base pair into another is called as a substitution mutation. The swapping of base pairs may be transition mutation or transversion mutation, depending upon if it is getting changed from pyrimidine to pyrimidine or pyrimidine to purine. Okay, transition is substitution of one pyrimidine by other pyrimidine or one purine by other purine. While in case of transversion, what happens is one purine is replaced by pyrimidine or vice versa. So these are transversion mutations. Frame shift mutation is sometimes it might happen that during replication some extra nucleotide may get inserted on some nucleotides may not get copied. So if this happens, so it can lead to the shift in frame, the reading frame of this DNA polymerase, and then it can lead to the mutation. Okay. uh, Not the DNA, the frame shift in the reading of this, sorry, during the protein formation, which can lead to the frame shift, and it will read, it will shift the reading frame, and that's why it can read miss, there can be some the misadditions of some amino acids that can lead to synthesis of either a different protein or a faulty protein. So this is what frame shift mutation is. Spontaneous lesions, so naturally occurring damage to DNA are called as the spontaneous lesions. That can be due to some deamination, that can be due to some oxidative damage. It can be anything which can lead to these kind of lesions, and it can lead to the mutation. Sorry, transposition is the transposons is a DNA segment that have the capacity to insert itself at any location in the genome without having any relation with the target gene sequence. They can get inserted anywhere in the genome. These are also called as the jumping genes. Okay, so consequently it can cause loss of gene function or gene appropriate inappropriate expression, over expression, or least expression, or no expression. Anything can happen. Now, the induced mutation, in which the mutation is caused by some external agent. It can be a UV radiation, it can be an ionizing radiation, it can be heat. All of these are comes under physical mutagens, which can actually cause a damage to DNA, and which can lead to the point mutation or mutation in the genome. Next is the chemical mutagens. The chemical mutagens can be base analogs. The best example for this is the bromo uracil. Okay, so it is actually the analog to thymine. So if which can lead to the obviously mutation. Then the deaminating agents are also there, alkylating agents are also there, intercalating agents are also there. If you guys, if any of you are from the biology background and has read about ETBR, ATBR is an intercalating agent, and it can cause mutations. It can cause deadly mutations. So, there are many systems for repairing these numerous unintended lesions or unintended mutations. There are systems for that also, but to repair this damaged DNA, so the DNA repair mechanisms can be single-stranded break if there is single-stranded break, and there are there are two types of breaks that can happen during the DNA repair. Uh, DNA damage, that can be single stranded break or double stranded break. If there is single stranded break or single stranded mutation that happened, so for that, direct reversal excision repair, a mismatch repair kind of processes comes into action. If double stranded break has happened, then homologous recombination or non homologous recombination is what can be seen. ठीक है, तो सिंगल स्ट्रैंडेड ब्रेक के लिए डायरेक्ट रिवर्सल होता है, डायरेक्टली एक्टिंग ऑन डैमेज न्यूक्लियोटाइड कन्वर्टिंग बैक टू इट्स आइदर ओरिजिनल स्ट्रक्चर, और अगर एक्सीजन रिपेयर हो रहा है तो इट इन्वॉल्व्स द एक्सीजन ऑफ द डैमेज सेगमेंट ऑफ द डीएनए followed by the resynthesis of the correct nucleotide sequence by an enzyme named as DNA polymerase. DNA polymerase, we have read already that they work in DNA repair mechanisms. ठीक है, बेस एक्सीजन रिपेयर हो सकता है, उसमें न्यूक्लियोटाइड एक्सीजन रिपेयर हो सकता है. सो ठीक है. उसके बाद है mismatch repair. It detects the mismatch that occurs during DNA replication and then it repairs whatever is the mismatch. It will remove one of the nucleotides and will add the correct one there. We are not going into these details because sir has already taught them in the videos. It was just a recap for you guys to recapitulate. Okay. Sometimes the DNA damage is so dangerous that it can stimulate the cells to produce DNA repair enzymes that allow an immediate reaction to that specific DNA damage. So these are called as the SOS response in bacteria. These are responses of these are actually generated by the DNA in an answer to the repair, so that a repair mechanism can activate it and the DNA damage can be repaired. And the homologous recombination and the non-homologous recombination, as sir already told us, that it will be further taught in this in the lecture series. So yes, this was what you guys must have read in the last week. This was just an very superficial overview of what was there in the last week's lecture. Now we'll be moving towards this assignment questions. So as our practice goes, again I will be asking you one question and everyone will write the answer to those questions in the chat box. No one will speak up so that everyone can participate. So we'll be starting with our assignment questions and everyone please answer them in the chat box only. Okay. So yes, the first question is, plasmids in prokaryotic cells replicate independently as they contain what? Sorry, Bhavna, couldn't see your question. Now I've seen it. You are asking about which polymerase? I couldn't get it from the sign. You can spell the name. Okay, so answer to question number one is yes, it is origin of replication. Okay, it is origin of replication. You are asking about gamma. Yes, it is for replication of the mitochondrial DNA for the chloroplast DNA, the organelle DNA basically. Okay, question number 2. Replicon is a DNA segment of prokaryotes that what? Replicon. What is replicon? Question number 2. Answer everyone. Yes, it is. It is that part which undergoes replication. ठीक है? Okay. Question number 3. The AT-rich sequence or at the origin of replication is recognized by which enzyme? Question number 3 का answer everyone. Yes, it is DNA helicase. It is the DNA helicase which recognizes the AT-rich region. Question number 4. Only following can trigger the initiation of replication. Yes, what? Fourth answer is C. Yes, complete methylation. Only complete methylation can trigger the initiation of replication. If it is semi methylated, the replication won't be initiated. Okay. Next question. Following coat single stranded DNA to prevent it from rewinding into double stranded DNA during replication. What is that? Question number 5. Everyone answer. Question number 5. Yes, it is single stranded binding proteins. So that the single strand of the DNA can be stabilized, and then the process can happen. Otherwise, it won't be stabilized and it will wind upon itself. Okay. Next is the following prevents supercoiling at the region ahead of replication fork. What is that prevents the supercoiling? Yes, it is topoisomerase. Topoisomerase is the enzyme which prevents the supercoiling. Okay. Next question. During prokaryotic replication, following seals the DNA fragment gap in the strands. Question number 7, everyone. Yes, it is DNA ligase which actually seals the gap. ठीक है? Next is the strand. Sorry. The strand is known as. Just one. The strand is known as following, where DNA polymerase 3 synthesizes the strand from 5' to 3' direction continuously towards the replication fork. What is that strand where the continued replication happens? Question number 8. Bhavna, you are answering for question number 8. I think you need to reread the question. Read it again. The question is the strand is known as following, where DNA polymerase 3 synthesizes the strand from 5' to 3' direction continuously towards the replication fork. The key term here is continuously. Okazaki fragments are not continuous, they are small fragments. that are then further ligated using DNA ligase. So Okazaki fragments are not continuous fragments, and the strand in on which the Okazaki fragments are formed is the lagging strand. So the other strand on which the continued replication is happening is called as the leading strand. So the answer to this question is the leading strand. Okay, I hope you got the answer. Next question. Following remove the RNA primer of both strands by the 5' to 3' exonuclease activity during elongation stage of replication. Which enzyme does that? Question number 9. Yes, it is DNA polymerase 1. It is DNA polymerase 1 which does that. Okay? Just a moment, guys. 9th तक हम हो गया था 10th. Following acts as an initiator for rolling circle replication. What is that? Rolling circle replication. It is rep A. Okay. Answer is right. It is rep A. Question number 11. DNA polymerase gamma can be found in following where it participates in D loop replication. I've just answered this question for Bhavna 5 minutes back. Answer to this question is question number 11. Yes, it is mitochondria. Question number 12. The sequence between two origins of replication is known as. The sequence between two origin of replications is known as. Yes, it is replicon. Okay. Next question. During eukaryotic replication, one of the two strands is synthesized in pieces known as what? Question number 13. Yes, now the answer will be Okazaki fragments because these are the small pieces. They are talking about the small pieces in which the DNA, the replication is happening. Okay, question number 14. Telomerase adds telomeric repeats at the parental DNA ends using following as template for the synthesis. What is that? Question number 14. टेम्पलेट क्या होता है टीलोमरेज के लिए? Yes, it is self RNA component. ठीक है. Next is shifting of pro. Just a moment, guys. Yeah, shifting of proton from its original nitrogenous base to form its rare form is called as. What kind of a change is that? Yes, the answer is tautomeric shift. ठीक है. So the answer to this question is a tautomeric shift. So with this, all of the questions comes to an end. From assignment, I hope you guys didn't face any problem. Still, if you guys have any questions, you can ask me. I'm here only to answer your questions. Any questions you want, you can ask me. Any questions, anyone? If you guys have any questions, then you can ask me. Otherwise, you guys can leave the meeting. We're done for the day. Any questions, anyone? Ask me. Otherwise, you guys can leave the meeting. Okay, good night, everyone. We'll meet next week on Tuesday.